I just wanted to get started. Okay, it is my distinct pleasure to get to welcome Dr. E.A. Quinn to the Department of Anthropology today. E.A. is just a fantastic, not only established but up and coming in terms of some of her newer research projects. Investigator within anthropology, she has not only a strong grounding in biological anthropology, but I think also a really welcome biocultural perspective on a lot of her work. And that makes sense when you actually look at her background. She has her undergraduate degree was from Emory, and then she did her PhD at Northwestern with Chris Kazawa. And now she's at WashU. And in fact, EA is about to be going on for tenure. She's going to be submitting her materials for external review in April, so she'll be associate professor, no doubt, Dr. Quinn next year. Some of her most highly cited papers have actually been a really important paper with Chris Kazawa on developmental origins of adult health, and also a paper on field and laboratory methods in milk research. EA has worked with populations in Cebu, some comparative work in the United States, and you guys will be hearing today about work she does with Tibetans in Nepal. And I will let her take it away from there because she has a lot of great things to share. So, thank you.
Thank you very much, Kate, and the colloquium committee for inviting me out to talk about a topic that, well, frankly, I chase people down to get to talk about. So this is a fantastic opportunity. I did want to actually build off of one of the things Kate said about how I really approach human biology from a biocultural perspective. This is my collaborator on this project, and he is a cultural anthropologist at WashU. So we are walking the walk and talking the talk. And I've taught him the secret physical anthropology handshake. He can now do children's anthropometrics. Not always willingly, but he will do them. And then our collaborator in Nepal. So this is collaborative work that the three of us have really been working on over the last couple of years, really investigating human milk among high altitude and a comparative sample of low altitude Tibetans living in the Nubri Valley, Nepal, and Kathmandu. So when we start thinking about classic models of adaptation, high altitude is one of those that almost immediately comes to mind. If you're thinking, what am I going to teach Negro students about adaptation? You go, oh, high altitude, cold adaptation, Allen's rule, Bergman's rule. But it's one of those immediate kind of knee-jerk responses. And we know that there are actually quite a lot of different ways to adapt to living in high altitude. Cynthia Bell's described the Andean pathways of biological adaptation at high altitude. We know there's a Tibetan pathway. We know there's an Ethiopian pathway. That there are these discrete physiological responses, genetic responses, and selective pressures that have occurred in all of these high altitude living populations. Some of these physiological changes are summarized here: increased basal metabolic rates, higher levels of circulating nitric oxide, and increased blood volume and blood flow throughout the body, and particularly during pregnancy, this appears to be important. Subsequent work on infants at high altitude has found that when compared to low altitude populations, that is populations that do not have long histories at high altitude, birth weights of high altitude adapted populations are higher. We know there's actual structural changes to the placentas. And we know that high altitude infants are biased towards weight gain, often at the expense of length gains. We also know there are survival biases. The classic studies of this are among Han Chinese that have migrated to the Tibetan plateau. Mortality rates among the Han Chinese, despite greater wealth and resources, were for a very long time, and still continue to be, much higher than those of Tibetans. And during some of these early transitions were as much as 50% higher. But kind of the knee-jerk classic study of this is really from the Andes, where it took 200 years after European colonization before a Spanish child was born at high altitude and lived. So hopefully that gives you kind of a comparative perspective. That's actually why the capital of Peru is in Lima, is because women had to go down to give birth, otherwise the babies would die. So you can see these real kind of important pressures of high altitude on population migration, both recently and with a little bit more time depth. So one of the things we're really interested interested in is how do children survive at high altitude? So we know that children from populations with these long histories simply do better as infants. They are growing more compared to low altitude peers moving to high altitude. They're surviving more. However, they do tend to show patterns of stunting and higher frequency of stunting and wasting, and patterns of low weight for age Z-score and height for age Z-scores when compared to lowland populations living at low altitude. So there are some compromises. However, there's some counter evidence or a growing body of evidence that suggests that this may actually be an adaptive phenotype. So one of the things that we've really become interested in as part of this adaptation is the role human milk may play in high altitude adaptation. So if you're an adult living in high altitude, there's some pretty significant selective pressures going on, right? Decreased oxygen availability, frequent cold, chronic hypoxia associated with decreased oxygen availability, shortened growing season, decreasing nutrition. And these are going to be especially acute for infants who don't have some of the same capacities to thermoregulate, who have higher nutritional needs per gram of body weight, and who are going to be more sensitive to things like cold stress and hypoxia. However, we know that, as we said, infants of these populations are doing better, and one of the things that we've hypothesized may be that there may be particular characteristics of milk that may be associated with promoting kind of an adaptive phenotype to high altitude. So Libby Kogel has argued that the immature phenotype is under greater selection than the mature phenotype. And this may be particularly important for ecologies with these numerous, pretty severe stressors. But we haven't really quantified infancy and immature phenotypes well in these environments, nor have we really thought about the role human milk is going to play in adaptation, or really thought about human milk as a normal characteristic of human variation. Right? If you pull a human variation textbook off the wall and open it, I am betting you there's not a single word in there involving the words human milk together. Maybe there's something on milk drinking and lactose persistence, but there's no real consideration for the way in which variation in milk as a phenotype, may actually be important. So our investigations into milk as part of adaptation are both looking at milk as a phenotype, but also coupling that with the understanding that signals, hormones, cytokines, growth factors, nutrients, fatty acids, immune markers in milk may actually be important in the production of the infant and possibly even the adult phenotype. So that milk is serving these kind of dual roles. So to investigate that, we decided that the best way to look at milk as part of human variation and to investigate some of these questions about adaptation was to look at milk in highly stressed populations, and populations living in variable ecologies, such as high altitude. So that's kind of where Jeff and I found this project. And specifically today, we're going to be investigating two primary questions. Is there an association between milk macronutrients or energy, and is there an association between altitude and metabolic hormones in milk? Apparently the old version loaded, so I apologize. I realize now that the slide is grammatically incorrect. It's R. Whoops. Are there associations? Multiple associations, not single pathways. Subsequent slides are correct. So we're looking at these two sets of association. But before we kind of start investigating these questions, is altitude affecting milk macronutrients? Is altitude affecting milk hormones? I want to situate you with where we are in the world. Here is India, here is Tibet and China, and here is the small landlocked country of Nepal. If you've been following along in the news, you know Nepal's had a really horrific last year. There was an earthquake at the end of April. The earthquake occurred about right here. I've got a later map I can show you. Here's Kathmandu. And more recently, and I'm going, India has closed the border to Nepal. And this is essentially starving the country of food, gas, cooking oil. The public health situation in Nepal is deteriorating rapidly. They're running out of medications. And there's a lot of tensions going on along the border. So within kind of where's Nepal, where's Tibet, that's the situation going on in Nepal. Here's the Nubri Valley, and you can see here's Tibet. This pass right here is one of the primary footpaths into Tibet. It's the Larkhe Pass. So these are not Tibetans living in Tibet. These are Tibetans living about 15 miles over the border in Nepal. But we know from genetic data and family histories going back between 7 and 10 generations, and from self-identity as Tibetans and ethnic social organization associated with Tibetans, that this is a population of primarily Tibetan descent. So within Nubri, so we're building into this red dot now. This is the Nubri Valley. It is located on Mount Manaslu, the 8th highest mountain in the world. So we're in the Himalayas on one of the very high peaks. This is Manaslu. And these are the villages in which we are actually working. So we're conducting this research. And you can see their altitude. They're ranging from 12,500 feet down to 7,800 feet. And I'll walk you through these geographically. And this is Samdo, the highest village at 12,500 feet. Som at 11,515. Shala, this is a satellite village of Li at 11,000 feet. And Lo at 10,500 feet. Hopefully you notice there's a very sudden jump in greenery between these last two pictures. And this is traditionally in high altitude studies what we often call the cutoff between high and low altitude. This is 10,000 foot marker, or between low and medium altitude, where a lot of the selective pressures of high altitude start to dissipate. And these pictures were taken within a 3-week time period, if that helps to provide context to kind of the nutritional density, the kind of physical environment that's available. The village of Li at 9,500 feet, and the village of Prok at 7,800 feet. So this is kind of the primary scene in which this is taking place. And I'm sure, of course, now your next, and of course then we have a comparative sample of mothers drawn from Kathmandu. So as part of our question, we did not want to only look at the effects of altitude. We also wanted to look at the effects of just being Tibetan, right? We want to know if something's more under genetic control, if it's more an environmentally induced phenotype. So we recruited 66 mothers from urban Kathmandu. And yes, 4,800 feet is not low altitude, but it is incredibly hard to find a large concentration of Tibetans at low altitude, and this was pretty much the best we can do. So this is comparative sample. Now I'm sure you're asking me really important questions of how do you collect milk at altitude? How do you do human biology in situations like this? How do you collect a sample that has to be frozen immediately in these kind of rough looking areas, at least in terms of logistics? Well, here's my field assistant collecting milk at this exact moment. My field assistant, the mother, the neighbor's kids. So we just try and keep all the hands out of the sample, and the mother is hand expressing. While we were collecting, we went into the homes and we interviewed each mother. We collected a milk sample between 6 and 10 a.m. to minimize variation, and this frequently looked a little something like this: setting up to work, getting the baby ready. I'm setting up equipment, my field assistants are getting ready. Once the samples were collected, they were processed in the community from the collection vessels, frozen in these tanks of liquid nitrogen, which were then toted from village to village by porter, as there are no vehicles and the walkways are, this is a very wide one, most of them are quite narrow and there's no horse traffic. So no vehicles, no horse, everything's being moved by people. And it's about an 8-day walk from the villages to Kathmandu. And the villages lack basic, there's very little power. About every third village will have electricity, micro hydro, and there is there is not much infrastructure, so there are some communal composting toilets and there are some communal pipe stands, but that varies from village to village. So villages are not only variable in terms of their altitude, but they're also highly variable in terms of their infrastructure. And when we were doing this early work, there were actually no clinics in the community. There are now clinics in 4 of the major villages. So the sample I'm going to present today is from our pilot data. And we had Wintergren and Leakey Foundation support for pilot work in 2013. The reason I'm not showing you more recent data is that we have just started a birth cohort study, and we only have 25 mothers in the birth cohort study, so we're obviously not getting data back yet. But this was the pilot study. These will all be cross-sectional data, and we're moving into a longitudinal model now. So there are 112 infants, mother-infant pairs in the study. Ages range from 2 days of age to 3 years of age. And we did not exclude 3-year-olds who were breastfeeding because that's within what is considered normal breastfeeding range for the population. So we kept them in the sample. So let's investigate that kind of first question. Is there an association between milk macronutrients, or is there an association between energy and altitude? And if not, what other factors in the environment might predict these differences? So to start, here are the basic characteristics of the sample. This is what milk looks like. If you've never had the opportunity to stare at its gloriousness, this is a sample of milk that's been left out on the bench to actually separate out. Top layer is fat, and in our population, our fat was 5.1 grams per 100 mLs. Our sugar content, sugar being down here in the liquid fraction, was 7.2 grams per 100 mLs, and our protein was 1.23 grams per 100 mLs. This added up to an energy per 100 mLs of 81.7 calories. And all the slides moving forward will be color-coded, so sugar's always going to be red, proteins are always going to be yellow. If that helps you kind of keep track of these macronutrients. So what do we find with our initial question? Here I'm only showing data for energy and fat. Fat shown here, milk shown here. If you're looking at the slides, an altitude's increasing as we go across each graph. If you're looking at these images and saying, wow, you found nothing? Excellent reading of the data. Yes, we found no association between altitude and milk composition for macronutrients or energy. We were disappointed in this, but not really surprised. We kind of did not expect there to be any because there's a tremendous amount of literature suggesting that human milk composition is quite stable. However, it was worth asking, and we thought it was worth funding, so we asked. What did we then find predicted milk composition? The things you would typically expect: infant age predicted fat and sugar, and this is the direction of the association, and then non-significant if it was not significant. Infant age, maternal BMI, and birth order. Nursing frequency did not have an effect. Altitude clearly did not. And milk volume did not have an association with milk macronutrients. You're probably noticing that nothing predicts protein. This is, again, completely in line with what the majority of other studies are reporting. Milk protein appears to be incredibly robust and is rarely predicted by any maternal characteristic. So, essentially, what this slide is summarizing is that milk composition in ethnic Tibetans is exactly the same as milk composition everywhere else in the world. It's following the biological rules that have been observed. However, we were intrigued by one particular aspect of this, and that is that the milk values we were seeing, the milk fat, was actually fairly high, and it was quite interesting. So we first did a comparison between mothers living at high altitude in the rural Newbury Valley and mothers living at low altitude in Kathmandu. And as you can see, another graph that shows you that there's no difference. Love negative data. This is negative data. So there were no differences. And again, we weren't really surprised, but we were still intrigued with this really high fat. And to kind of give you some context for that, the mothers between the altitudes are all similar, and there's no difference between the high altitude. But when we actually compare Cebu, or when we compare Newbury to Cebu, and we compare Cebu and Newbury where we have exactly the same methods, both my data, they were collected identically, to other populations around the world that have used similar methods, we actually see something really unique kind of emerging from the data, and that is that the milk fat in Newbury is quite high. And this was really, really surprising how high this was. And we noticed that initially when we did the analyses. No differences by altitude, but a general shift towards higher milk fat. And it is in fact the highest milk fat ever reported in the world. And when you couple this with the collection methods that we used, it is likely that we are in fact underreporting milk fat. So there is something going on here with milk fat that is unique. And to just give you context, here's milk fat, here's milk sugar, here's milk protein, right where we would expect it to be. My colleague at the Smithsonian that actually ran the data for us said that fat protein for energy plotted exactly where it should for mammals. So no surprise there, it's just this, it's the milk fat. It's something interesting with the milk fat. And so what we did here was we calculated an average, a mean for all of those populations you just saw on the prior slides for milk fat and energy. And we used a statistical technique that actually allowed us to compare milk fat for all those populations and weight the number of samples in each population relative to the percentage, or to what that was going to contribute to the average, and compare that statistically to Newbury. And what we found was that milk fat in Newbury is statistically different from the combined average of all of those populations. It is not only the highest ever reported in the world, it is statistically different from that of other groups. Now, of course, statistics are wonderful, but are they actually telling us anything biologically meaningful? And that was very much our next question. Is this a biologically meaningful difference? So if you calculate this out over what the average infant intake would be across a day, it works out to almost an additional 10 grams of fat per day and about 114 extra calories. So this might actually be a fairly significant increase. It is at least what some of the projections of neonatal brain costs are. So of infant daily brain costs being added to milk. And of course, one of the follow-up questions to this then that we had was, what is this doing to mothers then? It's great to think about this as an extra benefit to infants, but milk is being derived directly from maternal metabolism, so this is going to have consequences for maternal metabolism as well. So, following on this, we were, as we said, very excited, but there was no association between any of the macronutrients or total energy and infant size for age, using weight for age, BMI for age, and height for age Z-scores compared to the WHO. So we had milk composition that appeared to be higher, appeared to be different, but it was not directly associated with the infant's weight for age. This, of course, then set us up for our subsequent questions, looking at hormones in milk. So, okay, we know this little guy is getting a lot of fat, probably a lot of energy. We don't know if he's mediating his intake across the day, if he's drinking less. It is incredibly difficult to get 24-hour milk volume measurements because you have a couple of options. You follow the mother and the baby around for 24 hours and you weigh the baby before and after every breastfeeding, which means that you're sitting in the home at night, staring at the mother, waiting for her to breastfeed so you can weigh the baby. In a co-sleeping situation like we have in Newbury, you can imagine that might be a little problematic. very difficult to get good reliable data. You can give the mothers or the infants doubly labeled water, and, well, you can give the infants doubly labeled, or you can give mother doubly labeled water and measure it in the infant's urine. However, mothers aren't always excited about drinking heavy isotope water, particularly when there's no word for heavy isotope in the language. And you need 14 days of baby urine. These babies don't wear diapers. So logistical challenges to that. So our volume estimate for what infants are doing is actually kind of a compromise here. But we continue to ask questions about what a part of milk may be targeting infant biology. And so we shifted here to these two questions, and that is, are there associations between altitude, maternal characteristics, and metabolic hormones in milk? And are there associations between metabolic hormones in milk and infant weight for age z-scores? So similar analysis to before, again, starting with altitude and then moving to other maternal characteristics and ending thinking about infant biology. So here are the data that initially really got us interested in this question. And from here on out, the coding is going to be blue is Kathmandu and red is Newbury. And this will go across all of the slides except one that the colors have done something weird on. But that should help you kind of keep consistently following who's who in terms of Newbury versus Kathmandu mothers. And these are the data that really got us interested in the question of infant growth. And that is, for infants in Kathmandu, growth is fairly stable over the first 3 years of life. They are tracking along about a z-score of 0. However, for infants in Newbury, z-scores are plummeting across the first year of life. Weight for age z-scores, height for age behaves similarly. And we were really, really interested in understanding how patterns like this may be affected by milk, may be associated with milk. Are we going to see protective effects of milk on z-scores as have been reported in better nourished populations? So to do that, we're looking at just 2 specific hormones. We have done other analyses. In the interest of time, I'm limiting our discussion only to these 2 hormones today, but we are putting out, we are finished doing research on quite a few other growth factors and hormones, as well as immune factors. There's a lot of immune stuff going on that's also not in our discussion today. And these 2 hormones we're going to discuss are adipokines, so they're produced primarily by fat cells in the body. There is mammary production of both of these hormones, so they're actually produced by the milk-producing cells in the mammary gland. And these are adiponectin and leptin. In the existing literature, adiponectin is associated with infant growth, particularly weight for age z-score, and has an inverse association. So more adiponectin in milk predicts decreased weight for age z-score and lower change in z-score over the first year of life. Leptin shows a similar association, more leptin in milk, less overall growth. So some pretty straightforward questions. There are now some reasons to suspect that these models may not be as straightforward as has traditionally been assumed in the literature, and we've just put out a paper actually challenging this simple assumption when you actually start to look at these associations in highly pathogenic populations, which we'll discuss in a little bit more detail when we actually have the data in front of us. So let's start with adiponectin. Start with the A. When we look at adiponectin and altitude, there was no association, to the point where it is really not worth continuing, per se, this paradigm of looking for altitude-related changes. What we see instead is that there are actually differences in the association between maternal body fat and the amount of adiponectin in milk between the 2 populations. Neither population, however, has a significant association between maternal body fat and milk adiponectin. Again, this is not really surprising. This is something that's been reported in prior studies. It seems only in very heavy populations do you see an association. And then when we look at this in infants, is there an association between milk-borne adiponectin and child growth? Here's where the question starts to get a little bit more interesting. There is absolutely no association between adiponectin levels in milk and infant weight for age Z-score in Newbury. There is a significant inverse association in Kathmandu infants. So two very different patterns going on in two very different ecologies in a genetically similar population. When we then look at the hormone leptin, here is maternal percent body fat predicting concentrations of the hormone leptin in milk. Again, red is Newbury, blue is Kathmandu. We see a very strong association between maternal body fat and milk leptin in samples in mothers living in Kathmandu. We do not see a strong association in mothers living in Newbury. However, both associations were significant. Again, this is completely in line with the published literature. These are the exact associations reported in the literature. And as we have discussed in some of our prior work, building off principles that Andrew Prentice laid out for looking at leptin during malnutrition, leptin is a starvation hormone. It tends to be more sensitive at lower concentrations than at higher concentrations. So there may be greater sensitivity down at these low end to changes in maternal body fat compared to at higher ends of the distribution. And since the mammary gland can't itself produce leptin, there may be non-adipose tissue production of leptin. When we look at this, looking at leptin and its association with weight for age Z-score, we again see a cluster of data with an ill-fitting, non-significant association. And this, in fact, was quite surprising because prior studies have predominantly reported significant associations between weight for age Z-score and the concentration of leptin in milk, both at the time point and for change over time, with higher leptin predicting lower weight for age Z-score. And again, the association in the literature is always negative. We have, if there was any relationship here, if it reached significance, it would actually have been positive. And the association in Kathmandu, again, not significant, but approaching significance, and at least a little bit more in line with what's been described elsewhere. So these are some really surprising results given the context of the population. And they actually ended up raising quite a lot of questions about what is going on here in this environment. Now, of course, there are some major limitations. We only have a single sample of milk, and we collected using gold and not platinum methodology. I did not really go into our collection methods. We collect a 10 ml sample of milk between 6 a.m. and 10 a.m. each morning. And the reason we do this is that prior studies have found that the 6 a.m. to 10 a.m. window is 94% accurate of a 24-hour pooled milk sample. And a 24-hour pooled milk sample is considered the platinum standard. That is, you collect every drop of milk a woman produces over 24 hours, you put it in a big container, you shake it up, you take out an aliquot, and you measure the fat in that sample. Now, from an ethical point of view, I think we can all just basically feel our skin crawling at the ethical problems with this. You are literally taking a child's food away for an entire day so that you can make a pooled sample. And in the past, traditionally, the child has been supplemented with formula. There is 0 formula in Newbury. Now, maybe in some instances, some formula might be beneficial because if the mother has insufficient milk production, they tend to give the baby, they just put the baby on a cow, which we can imagine is pretty stressful on kidneys and child development. So there may be a place for formula in Newbury, but we did not want to introduce formula into this population We did not want to put our whiteness on formula or our training on formula, and we did not want the risk factors that would come with formula feeding. So we were completely against collecting platinum standard samples. Gold standard, however, is if you sample between the 6 to 10 a.m. window, it is, as I said, 94% accurate compared to that pooled sample. So I'm okay with that. And that's why I said we may in fact be underestimating milk fat, is because we're using this second tier technique, or gold standard. And we also do not take a full mammary expression. Since bottles are not common in this population, we do 2-minute feeding. The infant nurses for 2 minutes. After 2 minutes, the timer goes off. We pull the very angry infant off of the breast, and the mother hand expresses a 10 ml sample into the plastic jars you saw that I had, and then the baby's allowed to nurse until finished. We put the baby on the scale before and after this, so we know the volume that's being transferred for each feeding. This is what we have found, what others have found, is the best way to collect milk in an ethical way in a low resource population. And we can even ask if some of these questions should come up in other populations like NICUs, where they often will take full mammary expressions. Although there's a movement in NICUs now to pulling out of feeding tubes samples. So it's sticking to the side. So some of the limitations, as I said, it's a single sample, and that means not only the milk sample is a single sample, but it's also point measurement of the child's growth. We do have infection histories for the kids, so we know if they were sick at the interview, sick a week before, sick a month before. We do not have longer health histories, so we do not know what other illnesses may be going on. We know for the low altitude village in our sample, there had been an outbreak of diarrheal illness 3 weeks before we arrived, and 3 infants had died. So we may have survivor bias in our low altitude sample. And that's certainly something that we are aware of. And then, of course, in terms of our questions of what's going on with nursing behavior, mothers are nursing 12 times a day, or at least that's what they're reporting that they're nursing. We're assuming that they're transferring the volumes we were measuring, which was about 80 mLs on average, and maybe a little bit more, maybe a little bit less for each bout. And so we can't rule out then that if the milk is in fact higher in fat, mothers may be compensating for this higher milk fat by reducing how frequently they're nursing. So infants are taking in less milk of higher caloric value than more milk of lower caloric value, and that the overall net energy gain of the infant is the same. We also cannot rule out that we're simply missing associations because we are looking at points in time. So, these are kind of some of the questions that came out of this data. Now, one of the assumptions that was going on before this study was that infancy at high altitude was more energetically expensive. We discussed this with a couple of you, that this was done by measuring an 8-year-old, measuring a 12-year-old, and then drawing a line to 0. So we don't actually have metabolic measures of infants and children living at high altitude. So we don't actually know what the metabolic costs of infancy are. We don't have metabolic measures of lactating women at altitude. We do not know if there's a seasonal component. The study was done during the months of May and June. We do not know if there may be seasonal variations such as decreased growth during cold season or during harvest season when infants may not nurse as much. We do not know if, and this is one of our ongoing concerns about how these studies have been approached more generally, is that if weight for age is just a really bad measure of infant body composition, and if the hormones in milk may actually be targeting specific tissues, and there's a group actually coming out of the University of Oklahoma, David Fields and Ellen Dimarath, who have actually demonstrated that leptin, adiponectin, and insulin are targeting different tissue types. So higher leptin is predicting more body fat but lower lean tissue mass. And they're doing DEXA scans of infants and comparing it to milk. Beautiful study. Not something I can do here, because as far as I know, they have not invented a solar-powered DEXA. The moment they do, though. So that weight for age z-score may be missing an enormous amount of the picture. We do not know what the potential costs of this to the mothers are. If milk is higher in fat and they are delivering a higher volume, that's a tremendous metabolic cost to mothers. And we know for Tibetans, because of their patterns of marriage, particularly polyandry, women are having higher birth frequency than is reported for other high altitude populations. You have increased birth frequency. You could have most women in this population will be pregnant or lactating for the majority of their adult life. And in our population, there's much less polyandry now. And then, of course, we don't know how mothers may be mediating these costs by early and frequent use of complementary foods. So these are all kind of questions that really came out of this research, trying to think about why some of these models may have failed. And then finally, what we're jokingly referring to as altitude 2.0 is, what is altitude actually indexing? Is it indexing hypoxia? Is it indexing changes in infectious disease? You saw that sudden shift in the pictures. Imagine what that's doing to bacterial growth and replication, right? We refrigerate food to keep bacterial growth down. So what happens when you have that very sudden shift in terms of pathogen load of the environment? Is it indexing nutritional availability? And one of the kind of cool parts about this study was it was one of the first studies of Tibetans that actually had a pretty severe altitude gradient that wasn't restricted simply to the Tibetan plateau and looking at a very high altitude sample. It instead allowed us to look from about a medium altitude to a high altitude sample and see some of this gradient actually occurring within the sample. So the questions we just laid out were enough that we decided that it was worth continuing to do research in this area. And last year we were awarded a 3 to 4 year years of funding from the National Science Foundation to actually investigate longitudinal growth in children at altitude, collecting detailed body composition data using thermal imaging techniques, so we can actually differentiate using thermal cameras what is white adipose tissue, what's brown adipose tissue, and what's bone. Very cool. And actually quantify if there's associations between milk hormones and body composition, actually following infants for the first 18 months of life, so that we actually know when stunting is occurring, and infants are measured every month, and collecting milk samples across lactation to look at changes in milk hormones. And then, of course, it's expanded, as these things often do, to now include microbiome measures and looking at parasite load using DNA tagging. Pretty much any time a new group at WashU goes, hey, you want to measure X, and tells me they have funding, I'm like, I will collect that for you. So we may even be getting into milk microbiome and gene expression in the mammary gland, because when the milk is produced in the mammary gland and the fat pinches off of the cell, it actually takes a little bit of the cell with it. So you can actually get DNA from that cell line in milk. Not to mention, if you haven't heard, there are stem cells, pluripotent stem cells in human milk, and that is the coolest thing ever. So we were approved on the 11th of March at the AAPA meetings. If you were at the AAPA meetings, you know I was not cognizant by Sunday, because I was the organizer. So I was no longer functional by the day we got the NSF award letter. But it was very good news. We were going to start May 1st. We bought our tickets. We were ready to go. We had our field assistants in the community. We were leaving on April 26th. And you know what happened on April 25th is Nepal suffered 2 major earthquakes. Just to give you context for where this happened, here's Kathmandu, here's Newbury, here's the next valley over, Sum. Earthquake number 1 was right here. Earthquake number 2 was over here. Here's Everest, where you had the landslides and lots of people killed. So you can imagine if an earthquake here caused that much damage in Everest, what it did here. Here's what it did in Kathmandu, shot from the same location. And here's what it did in Newbury. Landslides, bridges out on villages. And this kind of completely re-engineered the project, or required us to re-engineer the project. We could not get into the valley. We could not get into Nepal. And it really would have been unethical to start the research this close to a natural disaster. 70% of the homes in each village were destroyed. It ranged from 90% in one village to 10% in one, but an average 70%. This is, of course, a huge trauma for mothers. And now as we move forward with our birth cohort, we have a natural experiment of stress during pregnancy in our birth cohort, and we are seeing suppressed birth weights in our first 25 babies. They are not where the birth weights were 2 years ago. So that is also something we're now grappling with. And of course, obviously you can't move a tank of liquid nitrogen when you have to cross this river. Well, you can, but I don't recommend it. I've done it, I don't recommend it. So the project was actually put on hold. And we did not, instead of starting May 1st, we started November 1st. And this was our original plan. So this was the plan for our start on November 1st. This is what we are now actually doing. So we've increased the number of villages. We've increased coverage. We will be enrolling about 90% of the babies. We've left out the highest village because there is extensive damage to the roads, and it is not safe to go to that village right now. So we've instead concentrated on villages between the 7,800 feet, shown down here for Rana, up to 11,500, added on additional villages. We're expecting about 120 births over the next 18 months, hoping for about 130 for a final sample size of around 100 infants, factoring in mortality. One of the pieces of really good news to report is that the communities are rebuilt. The communities did not wait on government support to rebuild. I would say, with the exception of Rana, the communities were completely rebuilt. We saw one still damaged home in Rana, one home that had not been repaired. So the situation has changed. We do feel that we can be there and actually do research now. And there's tremendous community acceptance of the research and community investment. And we're making sure that the information we have, not only are you guys seeing it, not only are we publishing it, but that we're communicating it to the mothers and working with the health clinics and the nurses and the local NGOs to communicate this and actually start to quantify the environment. And if we can figure out where the risk factors are, work with the NGOs to do targeted interventions. And then finally, I'll just thank the Colloquium Organizing Committee for inviting me, our fantastic field team, who are in the field right now, still collecting data, the tremendous research team we have in the lab, our families that let us go and do this for 3 months out of the year, that let Jeff and I go and do this, of course the mothers and babies, and community members who are participating, and then the Leakey Foundation, Wintergren, and now the NSF for the project support.
Go for it.
So, I just wanted to double check, what was the timing of the gastroenteritis outbreak?
It was 3 weeks before we were there. So
This year?
no, it was April 2013.
Okay, so earlier today we were talking about secreted IgA, is that these samples?
So yes, that's these samples, yes.
Okay, so if you're seeing, so I can't remember exactly what the details were, we were talking about how there were unusual levels of a secreted antibody called immunoglobulin A, which is something that's generally gut produced by B cells, but is also something you see in in class switching when you have exactly that kind of an infection. So are you seeing patterns of that being associated with those
So we only had a very small sample from that village because the mortality rate was so high among the infants.
Okay.
And these were the surviving infants 3 weeks later. And it was just one village. It was just Proc that had the outbreak.
Okay.
Each village has its own water supply. And there was a pipe burst in the Proc water supply, and sewage got into it. And I mean, we know the exact sequence of events that happened. And literally, John Snow ghost map it as to the severity of infections going down the water pipe. It's one of those kind of horrific sets of data that the traditional healer was able to put together by the time we got there. So this is the milk of mothers. So the immune factors would be in the milk of mothers whose infants survived the outbreak.
No, I'm talking about, like, everyone's being exposed to this, right? Everybody's using the same water.
Only in that village.
Only
Yeah.
in that village, but well, I guess, I don't suppose you're sampling people who no longer have infants, so that's
Right,
different.
yeah.
But those women would have also been exposed, right? Whether
Yes.
they manifested symptoms or not.
Yes.
They should have circulating B cells or plasma cells for the next 3 to 6 weeks that are going to be class switched to IgA.
And we, the paradoxical thing was the secretory IgA in the milk from Proc was the lowest of anywhere. So they actually had very low levels compared to the women in Kathmandu and the women in the higher villages. So that's actually kind of one of the questions we have as to what's going on there. We have not unpackaged that. That's why I did not put it in the talk, is we're thinking we need additional immune measures to triangulate what might be going on. It's kind of one of those questions that we have, and then to find that the milk SIGA levels were the lowest in Proc.
And that's strange.
It's very strange. And the mothers were saying that the infants were healthy, the mothers were saying that they were healthy. We cannot draw blood from the mothers. There's taboos against drawing blood. We are going to work with the nurses about when the mothers come in for hemoglobin screening, if they'll collect filter paper blood spots for us. But we don't know if that can happen in the future. But there are social taboos against collecting blood, so we could not do it in order to be able to test that.
I was interested in the inorganic components of the milk, particularly the calcium and phosphorus. It's drawn out of the mother's skeleton along with the dietary intake of the mother. Have you looked at the calcium content of milk?
We have not. We have not looked at any of the mineral content. We do not have the facilities in my lab to do that. This was, whenever in leaky budget, we kind of ran out of money to do those analyses.
Okay. Well,
So they'll be done in future on future samples. The Smithsonian Institute can actually do them for us as part of their normal testing. But right now we do not have any information on that.
I'm looking at a report on the relationship between osteoclast inhibition and adiponectin, which seems kind of interesting because it would slow down the maternal loss of calcium from the skeleton, and that might have an effect on the rate of skeletal growth and stature in the children if they're getting less calcium to build their bones.
And one of the things that I did actually not do, and I really should have done, is actually put the leptin and adiponectin levels in the milk in context. The leptin levels are incredibly low. When we reported last year leptin levels in the Philippines, we said, okay, we're at 400 nanograms per ml. We're reporting the lowest reported at this point, and we're now reporting an even lower amount of milk leptin, and we're seeing a similar pattern with adiponectin. We just had a paper come out end of last year where we actually described the fact that in Cebu, the adiponectin levels in the milk are outside the range that's ever been reported for human population. And we know it's not just that we have something happen to our samples in Cebu. We're replicating it in Newbury. We know, we've heard from a group working in the Gambia that they are also seeing adiponectin levels outside the range reported in the literature. And the same group has published data from the Igalo and the Aida, the foraging group in the Philippines, leptin levels outside the range, and outside the range that's been reported up until we all kind of started looking in non-weird populations to use that wonderful acronym. And really what we think is going on with the milk adiponectin may be that we've simply been mismeasuring it all along, is that with the populations we've been looking at, we've been seeing a very narrow, elevated picture, and that if we actually start to look at the full range, there's some really interesting stuff going on. But we're going to need more data from non-Western populations, and populations living in highly pathogenic environments with this kind of suboptimal growth to start piecing that together. One of the other things we know goes on with milk adiponectin is that it's involved in gut barrier function, and the intestine is actually full of adiponectin receptors. So adiponectin may be important in terms of maintaining gut integrity as well. So more questions than answers, I think.
Can I ask something of very different scale or level? Why Tibetans? You're working in Nepal.
We wanted populations that we knew had long histories of adaptation to high altitude, and the kind of classic models are the Mara, the Tibetans, and
Would Nepali folks
so
be perfectly acceptable?
Nepalis don't typically live up in the really high valleys. You'll have other ethnic groups like Sherpas, or a couple of different ethnic groups, and we could have worked with those. Jeff already had this ongoing relationship with Newbury, so it really made sense to go and work there. We have been invited to a couple of other high altitude sites with different ethnic groups, and
In the same general area?
in the same general area and in the Andes. So kind of starting to think about putting together a comparative framework, the same way that's been done for a lot of the kind of early breastfeeding and high altitude questions, and even for some of the adaptation to high altitude.
That's very useful, but I'm also wondering, what do you know about these Tibetans? Did they recently move into Nepal? Or are they of Tibetan origin, like we say Italian, when it's Italian Americans, and then maybe several generations back? Could that be something you need to take into account?
So we know that the Newbury Valley has been inhabited for 700 years, and we know that the majority of individuals in the valley have come over from Tibet. There are family histories, and Jeff just finished his book on the population history of the Newbury Valley, now looking at the depopulation that's going on, but he's also looked at the early population and migration in and out. Up until China closed the border with Tibet, there was movement back and forth across the border, almost continually. So marriage partners were coming in. One of our field assistants' father is actually from Tibet and living in Sama. So there's been a lot of transborder migration.
Over several
Over
hundred years?
700 years.
Okay, so
And
it makes me a little bit more nervous, right? So for example, I know you're saying that this is Jeff's work, right? But I would want to do careful geological work, because what if there has been some intermarriage with other populations that could, I don't know if it would explain some of your data or not, but you know, people don't like to often admit children
well, we don't
who
certainly
come from a, shall we say, a suspect.
we don't have
relationship.
paternity, but Cynthia Bell's been collecting genetic data in the
Right.
community as well, and the genetic data certainly suggests that there's a large percentage of Tibetan ancestry. There is both written evidence in family oral histories and genetic data suggest that there has been migration up the valley and intermarriage, but she is still untangling the genetic data. But we do have some genetic data, so we will be able to investigate some of that a little bit more
I
clearly.
would, because I'm just sort of a suspicious person.
But it is certainly something we thought of, and we have actually had a journal tell us that we need to provide a pedigree, that everyone is actually Tibetan before we can say they're Tibetan. Which is really interesting in terms of just politics of high altitude groups. Because this group self-identifies as Tibetan, they have relics from Tibet that were smuggled over the border right before the border closed, and they are recognized by the Tibetan government in exile as a Tibetan community. So we've taken that as, okay, we can call them Tibetan. Genetically, the high altitude individuals, I know Cynthia says that they have a much stronger Tibetan signal or whatever than the lower samples, and the village names don't make sense in the lower sample. They're not Tibetan names. They're local names where Tibetan characters have been mushed together to make the right sound. So the word is meaningless in Tibetan. So that's some of the stuff that we're untangling. And there's now a linguist working in the area with the linguistic data. Cynthia's working on the genetics, Jeff's working on the migration history, and working in human biology. So we're coming at this from a lot of different questions. So I think we'll probably be able to maybe get at pieces of that, but I'm not sure we'll ever be able to completely unpack what's going on.
Thanks, this is fascinating, thank you so much. I have a question about the languaging of the ranking of the methodologies that you mentioned. Not only do they mirror sort of Western expectations and label out credit cards right that way, but is there any conditions under which a platinum standard would be acceptable?
Apparently, if you're a mother in Western Australia, and a lot of the early work in the 1980s was done with the platinum standard, even in Bangladesh, Thailand, there's some really impoverished conditions. And one of the major milk labs in Australia uses platinum when they're trying to get full expression. And they've got some amazing mothers who are not going to nurse, who generally the mothers have expressed enough that they've got express milk they can feed the babies from before. And the mothers will literally collect cereal samples every 10 mLs for some of these milk labs to analyze. So they know every 10 mLs what's happening in the breast across a day. And that is far, far more than I feel I could personally ask any woman to do. Maybe one day when I have a kid, I'll have that patience, but...
And can I ask where it came from? Who devised this?
Who devised the methodology?
ranking?
It has more or less developed organically in the literature, really based on a lot of the early studies being this kind of very aggressive model of collection, the idea being that you could only say anything about it if you knew everything that was happening across a 24-hour period. And some of the work coming out of the Hartman group in Australia has collected 24-hour milk samples every month. So these beautiful sequences of data that are actually kind of disturbing from an ethical point of view and are very much sponsored by Medela, wanting to understand the dynamics with plumbing. So this is another one of the situations where there is some interplay with industry. But I suppose in terms of using the data, Medela is slightly less evil than Nestle. Slightly. But and this also kind of came out of, as more and more anthropologists started doing this and grappling with these issues, we were really reluctant to ever take a full memory expression and feed it back to a baby. And kind of our internal conversations about, well, what are the methods out there in the public health literature that are acceptable and that are reliable and accurate, and how much accuracy are we comfortable giving up? Because when you collect during the day, it's going to impact the milk fat tremendously. And we collected for milk fat. That was the variable we targeted because it is the most variable across the day. So that is what we aimed our sensitivity to. We know leptin and adiponectin do not vary across the day in milk. They're constant, so we don't have to worry about targeting as much with them. We know protein's pretty stable. But if we were going to look at something like relaxin, we would have to be explicit about when our samples were collected because we'd actually be missing the daily peak. So there's a lot of these kind of issues, and what's the association with volume, for example? And there's no good marker in milk that we can use as a measure of volume the way you can with some markers in urine. If they ever figure out a way to do it, it will be right there using it. So, I mean, these are all kind of limitations and questions, and we try to be really upfront about them because this is such a new area for anthropology to be getting into, and having these kind of very careful, thoughtful conversations with the nutritionists, with the public health people, with the anthropologists, laying out these concerns, and really questioning this language. Why is this platinum? Why are we not saying that this is unethical? This is beyond an ethical limit. Well, maybe it's not, the way the Hartman group does it, where you have mothers that are excess producers. So they have milk they can give their child. You know, that's an ethical issue they have to grapple with. I know how I grappled with this in my situation, and that was I will lose a little bit of accuracy in order to do what is kind of ethical data collection.
Thank you.
Thanks.
Okay.